So, for decades, we've kind of just assumed that the human embryo was, well, this solitary genius.
Oh, absolutely. Like it just miraculously knows what to do.
Right. We picture this tiny, featureless cluster of stem cells somehow just knowing how to build a whole complex human body all by itself.
Yeah, completely isolated.
Exactly. But, I mean, what if the embryo isn't the grand architect at all? What if, for like the most crucial structural decisions of your entire life, the embryo was literally just taking orders?
Well, it completely flips our understanding of early development. I mean, we are talking about a fundamental paradigm shift in biology here.
Welcome to today's deep dive. If you're listening to this, you're basically getting a front row seat to the unraveling of one of the most, well, fiercely guarded secrets in human biology.
It really is.
We're exploring this groundbreaking 2026 article from the journal Cell, by a team led by Shen, and it's titled Reconstituting Human Primitive Street Formation Through Extra-Embryonic Cell Coordination.
Which I know is quite a mouthful.
It is a massive mouthful. But the mission of this deep dive is actually pretty simple. We are going to look at the support cells, the tissues that eventually become the placenta and the amniotic sac.
Right, the stuff we usually ignore.
Yeah, we think of them as just biological bubble wrap, just scaffolding that gets thrown away. But this paper proves that they're actually the foremen on the construction site. They're actively shouting instructions to the embryonic cells.
And to understand why this discovery is so monumental, we really first have to talk about the black box they're peering into.
Right, the timing of it all.
Exactly. They are studying a process called gastrulation. And this happens in the first 3 weeks of life, right after implantation.
And it's a huge deal.
It's the biggest deal. It's the exact moment a hollow ball of cells basically folds in on itself and establishes your fundamental body plan. Like it decides where your head goes, your tail, your front, your back.
It's the blueprint.
Yeah, it forms the 3 germ layers that will, eventually become every single organ in your body.
But observing gastrulation in humans has been, well, nearly impossible, right?
Oh, yeah. Completely restricted.
Because for those of you listening who might not follow bioethics closely, there's this thing called the 14-day rule.
Right.
It's this international ethical guideline that basically prohibits scientists from growing human embryos in a lab beyond 14 days. And guess when gastrulation starts?
Right around day 14 to 15.
Exactly. So right as the most important architectural event begins, scientists have literally had to just stop watching.
Which means, researchers have had to rely heavily on animal models like mice or on these spontaneous kind of chaotic aggregations of stem cells in a dish.
And those just aren't the same.
Not at all. They just don't capture the precise, spatial and temporal magic of what happens in a natural human womb.
Yeah.
I mean, we've basically been trying to understand a full symphony by just looking at a few isolated instruments.
Okay, so let's unpack this. How did Shen's team finally get to listen to the whole symphony? Because to prove that the environment those support cells is doing the directing, they had to fix a major flaw in how we study stem cells.
Right, the chemical soup problem.
Yeah. Normally, if you want stem cells to do something in a lab, you essentially have to dump this huge cocktail of external serums and growth factors onto them.
You're chemically bludgeoning them down a specific path.
Exactly. It's a very heavy-handed approach.
It is. And the problem with dumping a biochemical soup onto stem cells is that you introduce, well, a massive amount of unpredictable variables.
I don't know what's doing what.
Exactly. You can't tell if the cells are communicating with one another or if they're just desperately reacting to the artificial chemicals you literally poured into the dish.
So the researchers invented this thing they called the F4 co-culture system. And F4 stands for feeder-free, chemical inhibitor-free, growth factor-free, and serum-free.
A totally clean slate.
But wait, I have to push back here for a second. If you take away all the food and chemical instructions that scientists normally use to keep these cells, active, wouldn't the cells just sit there and do nothing?
You would think so, yeah.
Because it sounds like putting a group of strangers in a soundproof room, taking away their cell phones, and just waiting to see if they eventually get awkward enough to start talking to each other.
That silent retreat is actually the perfect analogy, though. By stripping away all that external biochemical noise, the researchers created a pristine environment.
So they could actually hear the conversation.
Exactly. They wanted to observe the pure, unadulterated chemical conversations happening directly between the extra-embryonic cells, the support cells, and the embryonic stem cells, which we'll just call ESCs.
Right.
If something happened to the ESCs in that F4 clean room, it couldn't be because of a scientist's chemical soup. It could only be because the support cells specifically told them to do it.
Okay, so the room is completely silent. Now we introduce the first guests. The researchers brought in AMLCs, which are amnion-like cells.
Representing the early amniotic sac.
Right, the fluid-filled membrane. And when they placed these AMLCs next to the embryonic stem cells in this totally silent room, the embryonic cells started expressing this crucial marker called T, or TBXT.
And I want to pause on that marker, actually, because T isn't just any gene.
No, it's a big deal.
In embryology, the expression of T is like the universal biological flare gun that signals gastrulation is starting.
Oh, wow.
Yeah, it marks the initiation of the primitive streak, which is the physical groove where the body plan actually begins.
So instead of calling it TBXT, we can really just think of it as the green light.
Yes, the absolute green light. And what's crucial here is the mechanism of how it happened.
Because there were no outside chemicals.
Exactly. The amniotic cells turned on this green light entirely on their own. The researchers tracked the molecular pathways and found that the amniotic cells were acting as an inducer.
By secreting stuff, right?
Yeah, they secrete specific signaling molecules, chiefly WNT5B and BMP4. And these molecules physically traveled over to the embryonic cells and triggered the pathways to start gastrulation.
So the amnion is basically the director on the movie set just yelling, action.
Exactly. But a biological system this powerful, multiplying this rapidly, it also really needs breaks.
Right, because if everything develops everywhere all at once...
You don't get a human. You get complete chaos. And that's where the second cell type comes in, the TSCs, or trophoblast stem cells.
And these are the precursors to the placenta.
Yes.
And their response was wild to read about. When they co-cultured the embryonic cells with these trophoblast cells, not only did the embryonic cells not start gastrulating, but the trophoblasts completely shut the process down.
Yeah, they induced a total suppression of the T expression.
Wait, let me make sure I have this right. The researchers even tried to force the cells to develop anyway, right? Like, they blasted the cells with a chemical called CHIR 99021.
Oh, yeah, CHIR is a heavy hitter.
Right. From what I read, it's an agonist that forces beta-catenin to accumulate, which is basically a brute force, totally unavoidable command for stem cells to turn into T-positive gastrulating cells.
It's basically a chemical sledgehammer.
Yeah.
Normally, if you drop CHIR onto embryonic stem cells, they immediately start developing.
But when the trophoblast cells were in the room, what happens? Does the trophoblast still stop it?
It completely neutralizes it. Nothing happens.
That is insane.
The trophoblast cells effectively block the CHIR chemical from doing its job entirely.
So it's like the amnion is the director yelling, action, da-da, but the trophoblast is this massive bouncer at a club just holding back the eager fans, maintaining absolute order.
That's a great way to put it. And, you know, what's fascinating here is the evolutionary logic behind that bouncer.
Why would the placenta want to stop development?
Well, in mammals, the early embryo needs to be shielded from extra-embryonic signaling to prevent premature development.
Oh, because it has to attach first.
Exactly. It has to establish a safe distance and make sure it's firmly implanted in the uterus before it starts building complex organs.
That makes so much sense.
And through secretome profiling, the researchers actually found that these trophoblast cells secrete an inhibitor called FSTL3.
And what does that do?
It acts like a biochemical sponge. It soaks up and blocks the signals that tell the embryo to develop. They literally create a biochemical safe zone, ensuring gastrulation only happens at the exact right time and place.
Okay, let's just pause here and do a quick recap for the listener, because we're building a pretty complex mental map today.
That's a lot of moving parts.
It is. So, if you're tracking the toolbox we have so far, we've got the amnion acting as the ignition switch, the green light, and we have the trophoblast acting as the brakes, the red light.
Right.
And the RNA sequencing data really paints a picture of this wrestling match because the researchers found over 200 specific genes for early organ development that should have been turned on, but the trophoblast bouncer just kept them completely repressed.
It's a very delicate, high-stakes tug of war.
Okay, so the amnion has started the engine, the trophoblast is acting as the brakes. But, I mean, a car with an engine and brakes still needs a steering wheel, or these cells are just going to pile up on top of each other.
Right, they need direction.
Yeah. How do the embryonic cells know where to go once they finally get the green light? They don't just emerge, they have to physically move outward to form the structure of the body.
And that brings us to the third crucial player, the extra-embryonic mesoderm, or XSM.
Which has historically been super hard to study.
Incredibly difficult. They are notoriously hard to keep alive and stable in a lab setting.
The paper notes they had to do some serious detective work just to figure out how to grow them. They had to dig into a spatial transcriptomics database from a natural human embryo.
Specifically a Carnegie stage 8 embryo.
Right, and just for context, a Carnegie stage is just a standardized way embryologists classify the physical development of an embryo, regardless of its exact age and days. And spatial transcriptomics is just incredible technology because transcriptomics looks at what genes are turned on, but spatial transcriptomics tells you exactly where in the 3D space of the embryo those genes are active.
It's like having a map of a city showing exactly which houses have their lights on.
Yes, perfect analogy.
And by looking at that map, they realized that natural XXM progenitor cells have a really specific preference for their environment. Like, they heavily expressed receptors for collagen type I.
Oh, interesting.
And they had highly activated TGF-beta pathways. So the researchers adapted their lab conditions to mimic that exact environment. They grew the cells on a collagen I matrix, added specific proteins, used inhibitors to block competing cells, and it worked.
They actually stabilized them.
They established stable XM stem cells that they could culture for over 100 days.
Which totally set the stage for understanding that steering wheel mechanism. Because when they stimulated the embryonic stem cells to become those T-positive cells in the presence of these newly minted XM cells, the embryonic cells didn't just wander around aimlessly.
You know, they underwent a massive physical transformation.
Right.
A process called an epithelial mesenchymal transition, or EMT.
Okay, let's break EMT down.
Sure. To understand EMT, think of a protein called E-cadherin as biological superglue. It keeps epithelial cells tightly bound together in a stationary, rigid sheet.
Like a cement foundation.
Exactly. But during this transition, the cells downregulate E-cadherin. They dissolve that superglue, and they upregulate N-cadherin.
So it's the biological equivalent of swapping out a cement foundation for a pair of roller skates.
That is exactly it. They break their connections, become highly mobile, and they actively migrate.
And here's where it gets really interesting. They didn't just migrate anywhere. They made a beeline directly for the XM cells.
Yes.
It was like those support cells were a powerful magnet or a GPS beacon, just pulling the newly formed embryonic cells outward. And they tested this against other cell types, right? Just to make sure it wasn't a fluke.
Oh, yeah. And the embryonic cells completely ignored the others. They specifically wanted to get to the extra-embryonic mesoderm. The extra-embryonic mesoderm is providing the spatial coordinates. It's guiding the morphogenetic movements, the actual physical shaping of the cells that are required to build the early embryo.
Okay, so we have the start signals, the stop signals, and the GPS navigation. What happens if we put all three of these foremen onto the same construction site, arranged exactly as they would be in a human womb?
Well, this is where the team utilized some brilliant microengineering to build what they call the disc-gastruloid.
The physical precision of this setup is just wild to me. They use PDMS stamps, which you can think of as microscopic rubbery cookie cutters.
Right.
And they use these stamps and a gel-like substance called matrigel to confine the embryonic stem cells into a tiny, perfect, flat disc. And this disc was exactly 700 micrometers wide.
And that 700 micrometer measurement is no arbitrary number.
No.
No, it's the exact natural width of a human embryonic disc at Carnegie stage 6 to 7.
Which is right before gastrulation begins.
Precisely. Because in biology, geometry matters just as much as chemistry. The cells need to be at a very specific density and distance from one another to communicate properly.
So they have this 700 micrometer disc of embryonic cells. Then, using a 3D transwell system, they hovered the amniotic cells, the director's yelling action directly above the disc, kind of like a canopy.
Right.
And finally, they surrounded the outer edge of the disc with a continuous ring made of both the trophoblast bouncers and the XM magnets.
They essentially built a complete 3D model of a pre-gastrulation human embryo, placing every single support cell exactly where it naturally lives.
And then they just let the F4 cleanroom do its work. No external chemicals, just the cells talking to each other in their natural geometric arrangement.
Exactly.
And if you're listening to this and wondering, like, why this matters for you, think about congenital diseases, think about early miscarriages. If we know exactly how these specific foremen direct the building of a spine or an organ, we finally have a map to understand why things sometimes go wrong in those critical first few weeks.
We are unlocking the blueprint.
We really are. And the results of this 3D model prove just how accurate that blueprint is.
Oh, definitely. At 72 hours into this experiment, down the exact midline of that 700 micrometer embryonic disc, a physical groove formed.
Wow.
The cells in that specific groove lost their tight connections, they started expressing the markers for gastrulation, and they began to migrate. They successfully recreated the primitive streak in vitro.
Let's do another quick recap of the choreography happening here because it is just stunning.
It is a beautiful biological ballet.
It really is. So the amniotic canopy above is raining down inductive signals, telling the disc to start. The trophoblast cells on the perimeter are pushing back, secreting inhibitors that prevent chaos and force the streak to remain perfectly confined to the midline.
Right.
And the extra-embryonic mesoderm on the edges is exerting that magnetic pulling force, guiding the migrating cells outward from the streak.
And, it wasn't just a physical resemblance either. They proved the chemical language was identical to natural development.
How do they do that?
Well, when the researchers introduced drugs to specifically block the FGF, WNT, or TGF-beta signaling pathways in this model, the streak entirely failed to form.
Oh, wow.
Yeah, these support cells are literally speaking the molecular language of human development.
They even observed anterior-posterior patterning, right? Like making sure the head goes at one end and the tail at the other.
Yes, which is critical.
Because in natural embryos, the primitive streak only forms at the posterior or the back end. The front, anterior end is protected by antagonistic signals. And the researchers showed that these exact same chemical gradients were functioning perfectly in their lab-grown model,
keeping the head region protected from gastrulating too early.
It's unbelievable.
But, establishing those axes is a monumental achievement. But the formation of the streak is really just the starting line.
Right, there's more.
The researchers wanted to know, is this disc-gastruloid just a very accurate static snapshot? Or is it a living movie?
Yeah.
Does it possess the potential for true extended organism development?
And to test that, they performed a highly delicate procedure. They carefully detached the embryonic disc from its microengineered scaffolding of support cells and transferred it into a suspension culture.
Basically letting it float freely.
Right, in a nutrient-rich medium for an extended period up to day 4.
They let it run to see what would happen once the initial instructions had been delivered. And by day 4 in this extended culture, the structure didn't just fall apart.
Yeah.
It spontaneously organized itself into a complex, asymmetrical, curved, multi-layered structure.
And the diversity of cells it created on its own is staggering. I mean, when they ran the single-cell RNA sequencing on this floating structure, they identified 14 distinct cell clusters,
which mapped beautifully to a natural Carnegie stage 10 human embryo.
To put that in plain English, it started forming the very specific layers that make up our organs. On one end, it built neural mesodermal progenitors, the foundation for a nervous system.
The early precursors to a brain and spinal cord.
Right. And in the middle, it laid down the markers for a primitive gut. On the outer layer, it developed early neuronal markers.
And this raises a critical point about the cascade of biological development, actually. All of this late-stage complexity happened spontaneously.
Because the setup was already done.
Exactly. The extra-embryonic support cells provided the initial setup, that initial architectural blueprint, during the first 72 hours. But once that spatial and chemical foundation was laid, the embryonic cells knew exactly how to execute the rest of the plan completely on their own.
But as I was reading through their day 4 data, tracking all these different germ layers forming, I noticed something happening in the anterior region. They found cardiomyocyte progenitors.
Heart cells, marked by ISL1 and CTNT.
Yeah. And these cells didn't just sit there inertly, right?
No, they didn't. They actually formed a cavity-like structure and began to exhibit rhythmic contractions.
Wait, really?
They literally started beating in the dish.
A beating bundle of cells that organized itself into complex axes, formed primitive guts and nervous system precursors, all because of the highly specific, perfectly timed instructions given to it by the support cells in those first 72 hours.
It is the ultimate validation of Shen and his team's central thesis. The embryonic stem cells carry the incredible potential for life, but the extra-embryonic lineages, the amnion, the trophoblast, the extra-embryonic mesoderm, they are the ones holding the compass and the map.
So to recap our journey today, we've completely dismantled the idea that the embryo is this sole isolated genius building the human body.
Completely.
We used to look at the placenta and the amniotic sac as just scaffolding that gets discarded when the building is finished. But this deep dive reveals they are the master foreman.
The amnion yells action.
The trophoblast controls the crowd.
And the XM provides the GPS coordinates.
Right, they coordinate the symmetry breaking, the cell migration, the entire biological ballet.
And by proving this in a totally clean, chemical-free microenvironment, this team has given us a completely new, incredibly reliable platform.
It's a game changer.
It really is. We can now study the very origins of human life and understand early developmental failures that were previously, just locked inside that 14-day black box.
Which brings me to a final thought for you to ponder. We started by talking about the solitary genius of the embryo, and we've seen how scientists can now recreate the earliest, most impossibly complex architectural steps of human life in a dish.
Right.
And they do it simply by placing the exact right support cells next to stem cells in the exact right geometric shape. It's a flawless cascade of timed chemical gradients and physical forces.
Mm-hmm.
So as we get better and better at recreating these exact conditions in a laboratory, where exactly is the line between a highly complex biological algorithm responding to chemical triggers and the actual spark of an organism?
It's a profound philosophical question, and it's one that this new era of advanced stem cell models is definitely forcing the scientific community to confront head-on.
Something to mull over until our next deep dive.